for crypto for crypto, which is old data from Jesse's group, and also for Toxoplasma. We do have some for Plasmodium. I don't believe, I'll have to look. I don't think we have them in the genome browser. We have a search that allows you to find the genes that have the transcription factor binding site in their upstream regions. But we don't have a track for it, because you can imagine the track could be very, very busy, because some of these sequences are very repetitive, and they could be found in multiple locations. And so depending on, because you'd be specifically looking for things upstream of genes and within a specific region. There probably is a way to put a track, but I don't think we have one. Yeah, it's a good question. But we do have some search in PlasmoDB for Plasmodium, and then we're working on, hopefully we'll be integrating more data from crypto and Toxo.
So when you find that track that we were looking at, that predicted transcription start sites in Toxo, if you find a transcription start site that is in a UTR, like way over from the ATG, do you believe that?
Yeah, remember, so it's a good question. So I'm sharing my screen right now. Remember this, all the data you're looking at here. So some of it is based on algorithms and complete predictions. Some algorithms are really good and they give you some nice data, and some are pretty crappy, right? That's all we have. They kind of work, but they maybe were designed for the human genome, and now we're applying it to a parasite, and it might give you okay results, but there are going to be issues with it. Same thing with all the data tracks, right? It's data. It's coming from an experiment, and just like if you ran an experiment in the lab, you're going to want to have a bunch of controls, and you want to prove the result you're getting in many different ways to be sure of it. And sometimes you're going to get some artifacts. And especially if you're looking at results that require you to come up with sort of a scale of, well, this looks like the intensity is X. That intensity, above that intensity, that would indicate a really good, transcription start site. And below that, there may be one there, but I'm not sure. And this is kind of this data, this transcription start site data. That's how it was when you initially generate this. You will end up with lots of potential transcription start sites, but then based on the depth of coverage for each of these sites, then and probably some other calculations, they come up with a value, and then they pick one that they feel like that's the, and not manually, obviously they're doing this automatically across the whole genome, and so they're then picking one that they feel like is the most accurate one to be the correct transcription start site. So sometimes that's wrong, right? And it won't be correct. The other thing you have to always think about with these experiments as well is what stage or what experimental conditions were used for a particular experiment when they collected the data, right? So for example, if your gene that you're looking at is a gene that's expressed only in a particular maybe pathogenic fungi, and it's expressed only in that particular stage of the fungus. And the experiment for transcription start sites was performed on a different stage. That's not the stage you're interested in. And this gene is potentially either alternatively spliced, or maybe it doesn't have it's not expressed in that stage. Then you're going to get results that don't match, your expectations, right? And so you have to sort of keep that in mind. If you're looking at if you're interested in genes that are expressed only in bradyzoid stages of Toxoplasma, which you're looking at right here, and this experiment was done, most of the experiments are done on tachyzoids, which are the fast replicating stage of the parasite, then the data will be biased towards that particular stage of the parasite. It doesn't mean it's wrong. It could be biased, and you may not have the full story. Okay. Let's see. Are there additional questions?
Yeah. I didn't see any. Jesse reminded us that of Oxford Nanopore data.
Yes, I will. I was going to show that as well. Yeah. Okay.
Okay. And Ellie does ask a question. Can you obtain the sequence from the genome browser for an intron or TSS? Yes.
Yeah. So what you can do in the genome browser is you can highlight a region, select a region that you want to download, and get that sequence. So there are a couple of, and the way to do that, it's a bit convoluted when you first do it. You're like, really, why do you have to do it this way? But it kind of makes sense when you think about it. So each of these tracks that you're loading is a track that's basically, it's representing, for example, this track is representing gene models. So this track is really just a graphical representation of coordinates, right? And same thing for many of these, is just graphical representation of coordinates. So even though when we look at this, we think, oh, gene sequence, there's a genome here, there's a sequence. Why can't I just, click here and download this sequence, right? Well, this track is not a sequence track. This track is a coordinate track that you're plotting onto this genome browser. So what you have to do is you have to turn on another track for the actual sequence. And so we can go in here and we can type sequence. I think it's called reference sequence. I never liked this name for this, but that's what it's called. And so it's called reference sequence. So you turn on this track. And so this track, you'll notice
your thing is frozen now. Yeah, we're not seeing.
My screen is frozen.
Oh, there we go. We're starting to see it now.
We're back. Okay, maybe there was a glitch in the internet connection. Okay, so you can see, let me scroll back to the top. So here we're at the top. You see the gene models again? Is that moving? Is it moving? Yes.
We're seeing you type reference at the moment.
Oh, wow. Okay, let me.
Now we're back at the gene models, although.
Okay.
You need to.
I'm going to stop sharing and share again. Sometimes you need to do that.
Okay.
So let's do that. And my screen, desktop 2. Okay, are we back? You see my gene models, hopefully?
Not yet. There you go.
Okay, weird. Okay, so one thing I'm going to do before, so I remember I turned on this new track called reference sequence, but it's all the way in the bottom. Whenever you turn on a new track, it goes all the way to the bottom of your view, and we have a lot of tracks turned on. So one thing I'm going to do is I'm going to click on the first track, my gene models. I'm going to click on it, and you'll see there's this arrow thing here. I'm going to click on the arrow, and I'm going to pin this track to the top. Okay, that's a very useful feature because now this track stays up here. And so now when I scroll, kind of nice because you can actually move up to whatever track you're interested in looking at and sort of compare it to your gene of interest. And so this is the reference sequence, is all the way at the bottom, and you can see, zoom in to see the sequence. Okay, so because this is at the nucleotide level, so obviously at this zoomed out level we're looking at, it can't display the sequence. So if we zoom in, either using this magnifying glass, or I can just select a region right here and zoom in very closely. Okay, so now you'll see this track loads and you'll see the sequence, both the 6-frame translation, forward, reverse, and also the amino acid sequence. Okay, hopefully that's displaying for everybody. Yes?
Yes.
Okay, perfect, thank you. Okay, so the question was, I'm going to zoom out again a little bit. Let's just zoom out maybe a couple of clicks. And you'll see this error here. That happens when we're loading a lot of data and we're so don't worry about it. It'll hopefully disappear once we reload the page. Okay, so let's say we want to, when you reload the page, unfortunately, this is an annoying feature, is that when you reload the page, the pin is not there anymore and you have to sort of reset things. But so let's say you want to download the sequence around this transcription start site. So either you can zoom in all the way to where this point is, right? And say, oh, okay, there's the sequence. And you can just say, well, I just need the, whatever, 6 nucleotides, so you can write them down. The other option is you can click on this highlight button up here. And then you can, let's see, hopefully this will work. So as you see, I'm clicking in my screen and I'm dragging, and you'll see it's highlight this region. So I highlighted this region around this transcription set. So now it's yellow, okay? All right, so let's say this is what I want to download. And now let's go down to the track. Remember, our sequence track, that's a track that contains the actual sequence data, right? The other ones will just contain coordinates for your graphics. And so I'm going to click here, I'm going to click on the arrow, and now I'm going to say Save Track Data. I'm going to click on that, and now I can choose whether I want to download the visible region, which is this whole region we're looking at, or the highlighted region, which is the one I selected, which is, it even gives you the size 345 base pairs. It's in FASTA format, right, which is a standard sequence format. I can either save it or I can view it. So I'm just going to view it, and here's my sequence. Okay. So that's how you would download it from the Genome browser.
Perfect.
We have other ways to download sequence, and if we have time, not today, but if we have time at some point or at the end, we'll be happy to show you that, because it's a very nice configurable sequence retrieval tool. So if you have, let's say, 500 genes and you're interested in only downloading the 200 nucleotides upstream of the transcription or translation start site for all 200 at the same time, you don't have to do it one at a time. You can actually input the 200 IDs and do it in one shot. And that's a very, very nice feature. Let's say you're trying to identify transcription elements or transcription binding sites or so forth in the upstream regions. Okay. I think based on sort of discussions I had in my group, we can do this in one or two ways. I thought maybe one way is to just walk through one gene and go through the details, and then if we have time and people are interested, we can pop up, one of the other genes from the other groups and discuss it in a similar manner. Does that sound like a good plan? Are people happy with that?
That sounds great.
Okay. And again, throughout this, feel free to unmute yourself and just ask a question, interrupt me at any time. I'm more than happy to be interrupted, or just type your question in the chat, and we will answer them. Okay, so we're looking here at the gene model at the top. And again, a lot of this will be a repeat of what Dave said, but this gene model, again, depending on the genome you're looking at, you always should consider, well, where are these gene models coming from? And often these gene models are coming from just a completely automated algorithm, and they're not perfect. Just like anything else, algorithms are not perfect, and they may get some genes perfectly correct, but other genes they will get completely wrong, right? And so you have to keep that in mind. Don't assume that just because there's a gene model there and it's part of the official annotation, that this gene model is a good gene model. And if you have a genome that has alternative splice variants as well, right? You can be almost 100% sure that not all the splice variants are represented for any gene that you're looking at. So it's always important to consider some real data that can support your gene model or not. And I can tell you from personal experience, talking to people at conferences, there are a number of people who've sort of been led astray by sort of taking the gene model blindly without looking at the underlying evidence, which there's a lot, as hopefully you've gotten to appreciate. Without, they didn't look at the evidence, and it turned out their gene model was actually wrong, and they spent a few months trying to clone a gene that was just the wrong gene model because the algorithm did not call it correctly. Okay, and the evidence was right there. If they just looked at the evidence, they would have been able to figure it out, which is always sad to see. So always look at the real evidence. So here we are. We're looking at this particular gene. Let's scroll down. You'll notice here that there's a release 65 transcript. So because this genome, I think Dave mentioned that this genome was updated. So you will see that the release 65 transcript will be available as well, so you can compare and see if there are any differences. So moving down, this RNA sequence evidence for introns. So again, without going through too many details, but I'm happy to discuss this further afterwards when we close off the session at 1 o'clock if people need to know more about this. But these little lines here represent intron spanning reads. Okay, so remember when you do your RNA sequence experiment, you're getting it from typically fully spliced RNA molecules, right? So these introns will not be sequenced, or most of them won't be sequenced when you do your sequencing runs. And then you'll take this RNA data, the short read data you're getting, and you're mapping it back to the genome. And wherever you get a peak of binding, that tells you there's an exon. And wherever you get a trough or no binding, that tells you there's a possibility of an intron. And if we look at the raw data, here's some raw data of that. You'll see here's a lot of a big peak here. So there are a lot of reads binding here. So that tells you there's transcription going on here. It goes down. So it looks like, oh, there's potentially an intron here. And then here's more transcript. There's another exon, and boom, there's another intron, and so forth. And you can keep going down this line. And you see the areas here where it's not going down to zero, right? Which may be suggestive of something else happening here in this region, okay? So because the reads are coming from spliced genes, so where there are no introns, right, the messenger RNA, then you're going to have some reads that are spanning this intron when you map them back to the genome, right? And that's where this evidence is coming from. So anytime you get a read that had to be split to map it appropriately to the genome, that's basically a read evidence for that particular intron, for the existence of intron in that region. And if you mouse over these, you will see that these particular ones, the darker ones right here, have really good support. They're all about 8,000 to 9,000 or maybe 10,000 reads that support each of these introns in this particular gene. So you can be very confident that probably this gene model here is accurate, right? It has really good evidence for all of the introns. You will notice, however, there are additional intron spanning reads for this particular gene, and they're not nothing. I mean, they're not like 0, or they're not just like 5 reads. This one has this particular intron spanning, this particular species of intron spanning reads. There are about 1,350 of them, right? And so by this logic, right, you can argue, well, there's at least 1,345 cases where this exon was skipped, right? Or not cases, but at least reads that support that this exon could be skipped in some cases, right? And so if I were working on this gene, I would sort of put down a note in my notebook and saying, this gene may have some alternative splice variants. I may be interested in looking at that further, at some point in your project. And if you click on any of these, you will get a table that shows you basically how much of the, what percentage of the most abundant intron this was. So this represents about 12% of the most abundant intron. So again, just signifying that it's not nothing that you're getting here. And each sample that is that was assayed from the different experiments is listed here with the reads that are coming from that experiment, right? And so in theory, you can take this table and look at it and try and determine whether there are certain samples or conditions where this particular exon skip is happening more frequently, right? And sometimes this is easier said than done, but you could take this table and sort it and take a look and see at the samples and find out if there is some interesting correlation. Regardless, I would definitely keep that as a note, as you analyze this further, so that you don't lose sight of a possible interesting thing to look at. Okay. So then these transcription start sites. So this is again, it's based on real data, but also there's some level of confidence that was assigned to these, and so these are the most confident ones from their experiment. So they're pretty good, but they're not always 100% good. And so in this case, you can take a look at it. This transcription start site looks like it starts with this. Maybe that was the evidence that was used. But also what's important is to, and I'm going to pin this one as well here. It's good to take a look at the other evidence, and you can see, well, here's the actual read data, and you can see that it is almost matching, where some of these reads extend out to, basically. So there are some species where this UTR goes out up to here to this transcription start site. So it's probably a reasonable transcription start site, but it doesn't mean that you may have other versions of this. I'm going to skip this prediction. This is just another prediction algorithm for gene models. I'll skip it for now. And I saw, Jesse, you had a note in there, but maybe I could finish this, and then maybe you can show your example, if that's okay. So just quickly, the ChIP and ChIP-seq data. I know in my group, at least, they benefited from a quick explanation of what those are. Did everybody go through these in their groups, or should I do a quick explanation?
Might
Quick
need to
explanation.
do a quick explanation. I think.
Okay. So ChIP and ChIP-seq are basically the same. The concept behind it is the same. You're taking DNA that's bound to a protein, right? And you're using an immunoprecipitation of an antibody to that protein of interest. And then somehow you're detecting where that protein was bound, what DNA was bound to that protein. So the detection method is what distinguishes ChIP and ChIP-seq. In the case of ChIP, you're using a microarray, just like the old GLASS microarrays that were used for looking at expression profiles. You're using a similar array, basically, to determine where in the genome your particular protein bound. And in the case of ChIP-seq, instead of using a microarray, you're using a sequencing, basically. So just like you would do RNA or DNA sequencing, you're just sequencing the molecules, and then you're mapping them back to the genome. And wherever you get a pileup of reads, that tells you that, oh, that's the region where the protein was bound. Okay. I'm not going to go through the details of the technique, but the bottom line is you immunoprecipitate your DNA bound to your, in this case, modified histone, and there are different antibodies to different modified histones, and then you detect where those modified histones were binding. Depending on a modified histone, it may bind to different parts of your chromatin. You can mark open chromatin, you can mark closed chromatin, it can mark transcriptional units, or depending on your organism of interest, it may mark regions where the hypervariable genes are located, right? There are all sorts of modified histones that mark your genome in different ways. They're basically, they will be your epigenome and give you this whole other layer of regulation and access to your genes. And so it's actually much nicer to view this in a zoomed out. So if I zoom out a little bit more, just because of the how wide these peaks are, you can see now that as you zoom out a bit more, these peaks are a little bit tighter and you can see where they're located. So as I was telling my group, I don't remember, I never remember which of these modified histones binds where, and sometimes it actually changes depending on the modified histone between species. It actually doesn't have the same exact binding pattern. But what's easy, or what you can do, is exactly what I did right here, is you can zoom out and you can look and see where these peaks are. So for example, I can take a look at this histone 3 with an acetylation at the 9th lysine, basically, histone 3 canine, acetylated canine. And you can see there are peaks, in different locations. And if I try and see where these peaks are, you can see, oh, wow, there's a gene going from right to left and a gene going from left to right. And there's this really big wide peak in between them. Let's move down a little bit more. Looks like there may be a small peak right here, upstream of this gene. Here's another peak upstream of this gene. There's another peak in here, like upstream of this gene. Here's another gene, and it looks like there's a peak right upstream of it. So, so without knowing anything about this modified histone, I can draw a conclusion that it looks like it's marking sort of the upstream regions of genes. Okay, maybe it's marking promoter regions, right? So we'll leave it at that. If I look at this data from the ChIP-seq data, this histone 3K4 methyl 3, you'll notice it has a similar pattern. So I'm going to make the same conclusion about this modified histone. I'm going to say this is marking sort of upstream regions. Now let's look at the other modified histone. Both of them are the same modified histone, this histone 3K4 methyl 1, in the blue and the orange. You see the peaks are a little bit more depressed. They're lower, but that's fine. I mean, we didn't show this, but you could actually go in and change the score here, and we can make this, I don't know, 50. Maybe it'll make it look better, right? And so now you can see the peak, because now our scaling is a little bit better, and so you can see this peak. It's just they're less, there's less, I guess, data represented in that area. But now what you can do is you can do the same thing and say, well, where do I see the peaks? And you see now there's a gene, and there's a peak covering that gene, okay? And now this one doesn't look like it has a good peak, but this gene looks like it has a peak. And move down, here's another gene, and it looks like it has a peak, right? So again, and both of these seem to be following the same pattern. So where there's a gene, you get a peak. Where there's no gene, you get a depression or no peak. Okay.
Yeah.
So
We
again,
have a 5-minute warning for this session
okay.
before we move on to Apollo. Yeah.
Okay. So without knowing anything about this, I would assume that this is marking transcriptional units. Okay, the actual genes themselves. And so that's then you can use this then in association with all your other data to confirm, whether the gene exists in that region and whether and if you're interested in promoters, you would then look at this type of data. And I'll maybe highlight one more thing. And Jesse, I'm really sorry I'm taking too much time here, but if you want to highlight something important, then we can probably still do it now, or we can do it after this next session. But this is data representing long read data, which allows you to take a look at this. And so we have different types of tracks that show you, so the track down here is the actual reads themselves, right? And so you can look at that. So you can see these are all in theory, all the species, I guess, of transcript that were sequenced. And then these are gene models formed from these. And if you're interested, somebody else like Stuart or Catherine may be able to go into the technique details a bit more, or maybe somebody else on the call who could do that. But the bottom line is you can generate transcripts from the long read data. And so, for example, this is the most supported transcript. There's only one, and it looks like it supports a gene model. So that's pretty good. But you can see here that there are other ones that didn't have, 5 more than 5 reads. They had less than 5 reads of alternative splice variants. And it would be interesting to see if some of these match these intron spanning reads, the ones that are a little bit that have lower abundance. And so if we look here, you may be curious to see, well, but do I have ones that match these exon skip polymorphisms? And it's possible that some of these exist there, but they're very low in abundance. Okay, so I'll stop here. I'm sorry, I took way too much time probably, but Jesse, I'm sorry if I did not leave you any time. Did you want to mention anything or show something briefly?
Can I show my screen quickly?
Yes, let me.
This was a great segue because it's nanopore. So
great, perfect.
So this was a gene that our group had from group 9, and I've started with a very small window here where you can see there's 3 genes annotated, 1, 2, and then you can see this very long UTR that has multiple exons from a ferredoxin gene up front. And you'll see that this is hypothetical, 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 and you can see that there's a lot of different predicted introns, including you'll see that the most abundant transcripts, there seem to be a bunch of small exons right here that are missing. What's so cool is if you go and you open the Oxford Nanopore data, you open what we'll call an annotator's nightmare. So these are individual Oxford Nanopore reads that if you look at this, some of these reads are suggesting, as are these down here, that these 2 genes belong together in the same transcript. There's a lot of Oxford Nanopore reads that run through. And you can see in the Oxford Nanopore, we're picking up a lot of evidence for these missing exons that we didn't see. So what does the RNA-seq data look like? If you scroll down further there, you can also see that there's just this kind of blur of red all across here, again, with quite a bit of RNA-seq evidence for some of the smaller exons, but not all of them. And so where RNA-seq can be incredibly ambiguous and you just don't know where something begins and ends, the Oxford Nanopore can give you a little bit more resolution, although here all you want to do is pull your hair out and go, I don't know which is right. And I think it's really important here that if you look at the high confidence model, where there's more than 5 reads supporting, there's nothing. This is an empty field right here. There's a lot of conflict and confusing area in this region. And you would really love to know that if you were trying to work on this gene or that gene. I'm done.
Excellent. Thank you.
Great. Thank you.
I'm going to stop the recording and